Hi everyone. Today we're going to be wrapping up the end of chapter 11 and we'll be talking about signal transduction and cancer. Okay, so signal transduction pathways. This topic is important because it relates to cell communication. So cells need to be able to give each other messages just in the same way that sometimes you need to tell a friend something. And so the way that cells communicate with each other is through sending chemical signals. And so one cell will send a signal molecule to the other cell, and that signal molecule will attach to the receptor protein of that other molecule, and it will start a signal transduction. A signal transduction pathway is a series of molecular changes that converts an original signal on the target cell surface, and it leads to a specific response in that cell. And so one cell that sends the signal to the other cell receives that signal and then eventually does some kind of function based off the signal that it receives. So this is kind of just the run through of the pathway. The start off, like we said, once the other cell sends the signal to the second cell, and then that signal gets received by the receptor protein on the membrane. And then this activates just a series of relay proteins, or just a sequence of proteins where one protein changes and that affects the next protein. And so it's like a domino effect where all these proteins are changing based off this original signal. Eventually, this will activate a transcription factor, which you'll remember these transcription factors are needed for transcription to start. And so once this transcription factor is activated, a certain sequence of DNA will be transcribed into mRNA, and that mRNA will lead into a new protein. And so this creation of a new protein will eventually do that function that the original signal called for. And so going back, that original signal here in the very beginning of this chain eventually leads to the creation of a new protein, which will allow the cell to do something different or new. So an example of this would be if you get a signal from another cell that says that there's a lot of glucose in the blood, then you're going to start creating proteins that will be able to capture the glucose and bring it inside of the cell. So it's kind of like the signal to let the cell know that it's time to eat. Okay, switching gears now into cancer. Cancer is something very serious, and it's something that we've touched upon briefly in class, but we're going to look at the genetic basis of cancer. There are two major genetic reasons that cancer occurs. One of them is due to oncogenes. Oncogenes are genes that cause cancer, and they usually cause cancer by accelerating cell division or increasing certain proteins that accelerate cell division. And so you can think of oncogenes kind of like the accelerator of a car. Oncogenes, it can press the accelerator really hard and makes the car go really fast, in the same way that it makes cell division just increase exponentially. The second thing that causes cancer are mutations in tumor suppressor genes. Tumor suppressor genes are good genes, and what they do is that they kind of help regulate cell division, and they prevent cells from from dividing too much. But when you have a mutation in these positive tumor suppressor genes, then it stops the tumor suppressor gene from working, and so this eventually increases cell division. So you can think of the tumor suppressor gene as like the brake for the car, and a mutation in the tumor suppressor gene stops your brake from working. And so when you have these two things happening where you have an accelerator, like the oncogene, and you have a mutation in your brake, or your brake isn't working anymore, your car goes super fast and goes much faster than it should, and your cells are just dividing exponentially. And so just to go a little bit more in depth with the oncogene, some of these oncogenes can actually be transmitted through viruses. And so there's just a lot of different ways you can get oncogenes. And actually a lot of these oncogenes occur by being mutated from proto-oncogenes. Proto-oncogenes are just normal genes. They don't have any bad effects on your body, but these proto-oncogenes are very vulnerable to being converted into oncogenes. And so we're going to talk about a few ways this happens. So how do these proto-oncogenes, the normal genes, become cancer genes? They're generally they're just any genetic change that increases the proteins that are that lead to cell division, or they increase the productivity of the protein. And so one pathway here where a proto-oncogene becomes an oncogene, there's a mutation that occurs, and that mutation inside of the sequence changes the protein to become really hyperactive, or just like very really, it changes the protein to be really, increases the productivity and thus increases cell division. Another mutation that can occur in the proto-oncogene is that genes get replicated, and so you end up producing multiple copies of that protein when you only are supposed to be producing one. And so it like kind of doubles or even triples the rate depending on how many multiple copies there are of the protein. And so now you have an excess of protein. Another possibility is this proto-oncogene. What happens is that gene moves its location to a different part of the chromosome, and in doing so, it is now controlled by a different promoter. And now this new promoter might code much more quickly than the previous promoter. And so now this gene, which is exactly the same, just because it's in a new position, will lead to many more copies of the protein being produced. So this first mutation changes the protein itself and makes the protein more productive. These last two mutations produce the same protein, but it just in vast increased quantities. And this is exactly what we just talked about, but written down. So if you want to write this down, you can pause the video and write these 3 different mutations down. And this is just a more in-depth look at the mutation of the tumor suppressor gene. So normally the tumor suppressor gene stops cells from growing too much, and it just leads to just regular cell division. Now, if you mutate it so that it's no longer functional, then the cells keep on dividing uncontrollably, and that eventually leads to cancer. And the important note here is that cancer is progressive, and so you generally need accumulation of multiple mutations that, one, convert proto-oncogenes to oncogenes, and also knock out the tumor suppressor genes. And then the more you have these mutations, where you have oncogenes and you have mutated tumor suppressor genes, that will eventually lead to full-blown cancer. And so this is why usually people don't develop cancer until later in life. They need a lot of these mutations to accumulate over the course of their lifetime. And you can see that you start off with just a few cells that are mutating that in a way that they shouldn't, but eventually you can actually lead to very severe forms of these tumors, which eventually become malignant, or which becomes cancer. And that's just because there's a lot of mutations. Okay, thank you guys so much for watching, and this wraps up Chapter 11 for our class.